So on last week when we were together, we were talking about DNA replication and a little bit of history on DNA. So if you recall, we saw this slide already, but this was sort of a review of what we talked about last time, which was the flow of genetic information from the organism. Inside the organism's many cells, we have the nucleus of our cells. Inside the nucleus of our cells is where the chromosomes are found, and chromosomes, of course, are made up of DNA. DNA is going to directly be related to the production of a different type of nucleic acid, which is RNA. The role of RNA is essentially to get that genetic information, get the code out of or the instructions, if you will, out of the nucleus of the cell, ultimately to the site of ribosomes, which is where proteins are made. So that's kind of what we're going to be thinking about today, is how that happens. So that process of getting the DNA copied from the DNA to the RNA, and then out of the cell, and ultimately to direct the manufacture of proteins in the cell itself, in the cytoplasm, or wherever we have ribosomes. That's what we're going to focus on. And of course, the information that's carried on the DNA is going to code for all kinds of heritable traits, right? And you can see on that picture all different sorts of things that the genes are going to determine, what kinds of phenotypes we see in our individuals. All right, so I believe this is the last slide we saw in class. Genetic information is flowing from the DNA to RNA in two steps. So the copying of the DNA to the RNA strand, the messenger RNA strand, is called transcription, and we'll get into the details of that in this video. Once that RNA leaves the nucleus and finds a ribosome, that message is going to be translated. So the second step in protein manufacture is translation. And that's again going to happen wherever there's a ribosome. And that's going to direct the production of all of the proteins that we produce, and ultimately affect the function of all of the proteins that cells can produce. So the first of the two steps, again, is transcription. So last week we talked about DNA replication and how that happens, and it's going to be similar, except for what we're going to do now is we're going to copy that message from the DNA to a single RNA strand. So that's what transcription is. Transcription is following the DNA base pairing rules, which we talked about last time, but instead what's going to happen is we're going to make one substitution of one of the bases. So instead of thiamine, we're going to use uracil. So DNA and RNA are very similar. They're both nucleic acids. DNA is double-stranded. RNA is single-stranded. DNA has the base thiamine and a different sugar. The sugar for DNA is deoxyribose. RNA has the base uracil instead of thiamine, and instead of deoxyribose as the sugar, it has ribose. But transcription is the copying of the information from the DNA strand to the RNA, single-stranded RNA. So the DNA has to unzip again, right? But this time, instead of unzipping the entire DNA molecule, we're just going to unzip a portion of it and copy that DNA to the RNA strand. And again, what the ultimate goal here is to direct the manufacture of protein, because this is what makes all of us unique. So there's two stages in this process. Once again, transcription is the one you see on our slide here. Transcription is going to be very similar to DNA replication. And then the next one we'll talk about is translation. So the message that gets copied from the DNA strand gets copied to a type of RNA, which is called messenger RNA. And the important thing to remember is where this happens. And this happens inside the nucleus of the cell. The role of that messenger RNA is to get that information out of the nucleus and ultimately to a ribosome, where it can direct the production of proteins. Okay, so a good analogy here would be like, I think we use this analogy in class, would be like a cookbook. So the DNA is the whole recipe book, right, inside the nucleus. But if we just want one recipe, we'd copy down that recipe and then ultimately cook that recipe based on the instructions that we copied. And that's what translation is going to be. All right, so let's look at this again. Here's our picture again of transcription. The important thing to remember is it's creating a molecule of RNA from a molecule of DNA. And again, the name of that kind of molecule is messenger RNA. So let's see if we can get this to show us here what's going on.
Is cramming for her biology. Frosting covered for lunch. Specialized cells respond to the amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells. Manufacture. Instructions are coded by in transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so that was both transcription and translation and all the details. So transcription, what I want you to remember is where it occurs. So that occurs in the nucleus, and it's again the copying of the message or the code from, or the instructions, I guess is a better term, from the DNA to the RNA. When that message leaves, it's going to leave through via the messenger RNA through the nuclear envelope, and it's going to go and it's going to wedge in through the 2 subunits of the ribosome. And then from there, that message is going to be read, and another type of RNA called transfer RNA is going to go and gather the appropriate amino acids based on the message that it's reading. So that's the production, and it's going to bring those amino acids back to the ribosome, and then we're going to link them all together, which is going to be the making of the protein. So again, the thing you want to remember about translation is it's going to involve 3 kinds of RNA. The messenger RNA that we made in the nucleus, the ribosomal RNA, which is going to be out in the cytoplasm or stuck to the endoplasmic reticulum, and finally the transfer RNA, which is responsible for basically assembling the appropriate amino acids and bringing them back to the ribosome for protein synthesis. So this big purple thing is the ribosomal RNA, and the messenger RNA is kind of wedging in there. And then this gadget here is the transfer RNA. So the transfer RNA essentially has 2 ends that are important. The end that the amino acid is going to stick to, and then this end right here, we call this the anticodon, which lines up with the codon, which is the 3 bases in a row on the messenger RNA strand. So when we think about the genetic code, we say that the genetic code uses triplets. So the language of the genetic code is really read in nucleotide sequences, 3 nucleotides at a time. So the 3 nucleotides at a time is referred to as a codon. So basically, in that messenger RNA strand, every 3 bases is a codon. And each one of those codons matches to one of 20 amino acids. One end of the transfer RNA, the lower end down here, this part is called the anticodon, and the top end is the part of the transfer RNA that the amino acid can be stuck to. So basically, the transfer RNA has an anticodon, which is going to match up with a codon, and that's going to basically tell us which amino acid that we need to grab. So this is kind of how it looks again. This is the order of amino acids encoded by DNA, which of course is in the nucleus. So here's the DNA strand. We copy it with the complementary base pair, so adenine, uracil, guanine, cytosine, thymine, adenine, and on you go. So the 3 in a row is called a codon. These 3 amino acids, these 3 nucleotides, bases are going to be representative of an individual amino acid. So these are the amino acids. So the instructions come from the DNA. They get copied to the messenger RNA. That's transcription. And then out in the nucleus, they get translated into an amino acid sequence, which you can see here. And those amino acids are going to continue to be linked together until we get to the end of the message, at which point there's a codon that would be a stop codon, meaning stop read here, cuts it off, and there's our protein. So this is something you're going to see this week in your lab as well, if you haven't already done so. This is a way to kind of like figure out what the appropriate amino acids is based on the genetic code. So how you would figure this out is you have your first bases here, GACU, second base in the codon up here, and the third bases of the codon over here. So let's say we have AUA. So we'd find A here, then we'd find U here, and then we'd find A over here. AUA is going to tell us that we need isoleucine as our amino acid. CCC would be proline, CAA would be glutamine. You get the idea. There's a couple interesting starts and stops. You might be thinking, like, how do they know when to start reading? Well, when they get to a start codon, which is always AUG. And then they'll continue to read that strand of messenger RNA until you get to a stop codon, and you can see there's 3 different stop codons. Any one of those would stop the copying, stop their linking of the amino acids. You see there's a lot of redundancy. We only have so many amino acids, 24 to be exact, and we have all these different possible codons. And so you can see, like, for example, leucine, there's 4 different possible, or actually 6 different possible codons that would all be read as leucine. So there's a fair amount of redundancy here. Okay.